In the ongoing effort to extend not just lifespan but health span, the number of years a person lives free from disease and degeneration, scientists are increasingly focused on the invisible battle within our cells. One of the most important frontiers in this battle is the gut, where trillions of microbes, immune cells, and intestinal linings maintain a fragile balance that can be disrupted with age. Now, a powerful new study from the Buck Institute for Research on Aging offers a compelling glimpse into how we might protect this essential system as we grow older. By identifying and inhibiting a single enzyme, ADAM19, researchers have found a way to reduce gut inflammation and tame the harmful behavior of aging cells, pointing the way to treatments that support longevity without resorting to cell destruction. The discovery centers around a class of cells known as senescent cells, cells that have stopped dividing but continue to remain metabolically active. Though they play roles in wound healing and tumor suppression, their persistent presence in the body over time is increasingly viewed as a driver of chronic inflammation and age-related disease. This happens through their release of inflammatory compounds known collectively as the senescence-associated secretory phenotype, or SASP. Over time, SASP contributes to tissue damage, metabolic dysfunction, and a host of age-related disorders, particularly in delicate and fast-renewing tissues like the intestinal lining. But here's where the Buck Institute team's discovery shines. Instead of killing these senescent cells, as some emerging senolytic drugs aim to do, they have developed a more refined approach. By inhibiting ADAM19, a metalloprotease enzyme that regulates how SASP proteins are released, the researchers effectively silenced many of the harmful signals these aging cells send. The result was a noticeable drop in inflammation and gut permeability in animal and human cell models, achieved not by destroying aging cells, but by calming them. This method represents an important philosophical and scientific shift in the way we think about aging at the cellular level. Rather than declaring war on aging cells and wiping them out, this new strategy embraces a kind of cellular diplomacy, preserving the cells while stopping them from behaving destructively. It's a gentler and potentially safer alternative to more aggressive interventions, and it reflects a more holistic understanding of the cellular microenvironment. The research team's approach was methodical and comprehensive. First, using fruit flies, a model organism with surprising genetic similarity to humans, they identified the ADAM19 equivalent, a gene called meltrin, as a significant factor in gut inflammation after radiation exposure. When this gene was suppressed, the flies displayed stronger gut barriers, lower inflammation, and fewer signs of cellular aging. These findings were then confirmed in mice, where the chemical inhibitor batomastat was used to reduce ADAM19 activity. Once again, the treated animals showed improved gut integrity and reduced inflammatory markers following chemotherapy. And most importantly, the results translated to human cell cultures, where ADAM19 inhibition led to lower expression of key aging markers and inflammatory proteins. What makes these findings so exciting is how elegantly they connect gut health, aging, and inflammation in a single therapeutic strategy. Aging is a complex, systemic process, but this study shows that targeted interventions, even at the level of a single enzyme, can have broad and beneficial effects. By reducing the intensity of the SASP, the researchers not only preserve the integrity of the gut lining but also lessen the chemical stress signals that contribute to broader tissue damage and immune dysregulation. This holds particularly powerful implications for people facing chronic gut conditions, such as inflammatory bowel disease or the long-term side effects of chemotherapy and radiation. These conditions, which often worsen with age, are difficult to manage because they are driven not just by immune dysfunction, but by the very architecture of aging cells. A treatment that can quiet these cells and restore the body's ability to maintain gut health could dramatically improve quality of life for aging populations and potentially extend healthy years by slowing down systemic inflammation. Another key strength of this discovery is its precision. Unlike many anti-inflammatory drugs that suppress the immune system broadly, increasing risk of infection or cancer, targeting ADAM19 is more specific. The enzyme is involved directly in the release of certain SASP proteins, and its inhibition reduces the harmful output of senescent cells without disabling their other beneficial functions. In other words, this therapy doesn't take a sledgehammer to the immune system, it uses a scalpel. From a wider aging science perspective, this work represents a maturing of the field. We're moving beyond simply identifying that aging is bad and toward nuanced, mechanistic insights about how aging damages tissues and, more importantly, how to intervene at critical molecular points. The ADAM19 study offers a powerful demonstration of this evolution, providing a roadmap for similar strategies across other organs and systems. As researchers continue to explore this pathway and refine ways to deliver ADAM19 inhibitors safely in humans, the potential benefits stretch far beyond the gut. Chronic inflammation is a hallmark of aging not just in the intestines, but in the brain, the skin, the arteries, and nearly every system in the body. A well-tolerated xenomorphic therapy, one that modulates aging cell behavior without killing cells, could be a cornerstone of next-generation longevity treatments, keeping people healthier, longer, with fewer side effects than current interventions. In a world where age-related diseases threaten to overwhelm health systems and shorten lives, this research offers a rare and welcome piece of good news. The human body, with all its complexity, may still be vulnerable to aging, but it is not helpless. By identifying the right molecular targets, like ADAM19, and intervening precisely and respectfully at the cellular level, science may yet help us age, not just more slowly, but more gracefully.